Okay, so first of all, I would like to thank the possibility to present our results at this conference. So I would like to start that. So the MHC is, as we know, plays a central role in immune responses. They have essential role in maintaining immune homeostasis by interacting with immune cells via a plethora of mechanisms. For example, via the cell surface associated molecules or soluble mediators, which could be stored in microvesicles or exosomes as well. MHCs are not spontaneously suppressive cells. Their immune modulatory role depends on the actual environmental conditions. There are several strategies for immune modulation and therapeutic use of them. Their stimulation by inflammatory cytokines, for example, different kinds of cytokines, growth factors, or toll-like receptors, is an inflammatory cytokine licensing process for the MHCs. During an immune response, the T cells, antigen-presenting cells, and other tissue-resident cells produce inflammatory cytokines, leading to the release of immune suppressive factors, altered expression of surface molecules, and, for example, the production of growth factors by MHCs. During our work, we used the mesenchymal stromal cell line. Those were the MHCl cells. And these cells, as we know and as we proved that in a collaboration project, fit the criteria defined by the International Society for Cell and Gene Therapy. They are adherent cells. They express the MHC markers like CD29, 44, 73, 19, but don't express hematopoietic markers, and they are able to differentiate into bone, cartilage, and fat cells, so they have multipotent differentiation potential. And finally, they have strong immune modulatory capacity. In this work, we investigated the direct and indirect effect of MHCl cells on the differentiation of monocytes into dendritic cells. Dendritic cells are the most potent professional antigen-presenting cells capable of initiating adaptive responses and tolerance induction, maintaining and restoring the sensitive balance between the tolerance and immunity. So in this project, our question was that does MHCl cell line modulate the differentiation of dendritic cells from monocytes? So in our experimental setup, freshly isolated monocytes were differentiated into dendritic cells in the presence of IL-4 GM-CSF alone, or with these cytokines and MHCl cells or MHC-derived conditioned media. After the differentiation process, the protein producing profile, the cytokine production, and the T cell polarizing effect of MODCs were observed. First of all, to analyze the immunomodulatory effect of MHCl cells, MODCs were differentiated in the presence of MHCl cell-derived conditioned media, as you can see that in the bottom of the heat map, or in the presence of MHCl cells. The presence and the levels of soluble mediators in the cell culture supernatant were monitored by a protein array. Without the details, we can see that we identified factors which production was similarly up or down regulated by both MHCl cells and MHCl conditioned media. However, we have found soluble factors, which factors secretion was differently regulated by MHCl cells or MHCl-derived conditioned media. These results demonstrate that the baseline levels of both inflammatory and anti-inflammatory mediators in MODC cultures can be altered by MHCl cells in direct and indirect ways as well. Human monocyte-derived dendritic cells, so the MODCs, can take up and present antigens to T cells and can either activate or inhibit them through the regulated expression of co-stimulatory and co-inhibitory molecules. However, as it is visible, the communication between the APCs and T cells and the co-stimulatory molecule patterns are very complex, more complex than is visible on this picture, of course. Naive T cell activation by DCs requires two signals, termed signal 1 and signal 2. Signal 1 is equivalent to the binding of TCR to the peptide MHC complex. Signal 2 requires the interaction of co-stimulatory molecules at the interface between DCs and T cells. The onset of strong adaptive immunity crucially depends on the activation of dendritic cells. Positive red and negative blue signals have been identified and is visible on this picture or figure. So, after this, in this present work, we focused on the co-inhibitory molecule CTLA4, which molecule traditionally is expressed by lymphoid cells to limit the co-stimulatory potential of myeloid APCs. Basically, CTLA4 can prevent the binding of CD28 receptors to CD80 and CD86 ligands, resulting in the negative regulation of effector T cell responses. Interestingly, and what is extremely important in the regards of our project, an increasing number of studies demonstrate the expression and production of CTLA4 protein in different myeloid lineage cells, indicating that it could be a possible target of the MODC-mediated immune regulation. So to get insight how the presence of MHC-CM, so conditioned media or MHCl cells, affect the phenotype of MODCs, we observed the expression of cell surface molecules by flow cytometry. And as you can see here, based on our observations, the MHC cells significantly increase the expression of CD80 and PD-L1 antigens on the surface of the MODCs, while the cells significantly and additionally MHCs significantly increase the expression of CD86 and surprisingly, exclusively just MHC conditioned media was able to significantly increase the expression of HLA-DQ and CTLA4 molecules on the surface of the monocyte-derived dendritic cells. Interestingly, when the immatured but already differentiated MODCs were exposed to MHC conditioned media for 2 days, no or just minor alterations in the levels of co-stimulatory and co-inhibitory molecules on MODCs could be observed. These results suggest that MHCl-derived secreted mediators affect the generation of immune regulatory MODC phenotype only at the initial stage of their development. Thus, our next question was that what kind of factors are present in the MHC conditioned media determining the differentiation of MODCs in its initial phase or steps. As we know, MODCs could differentiate toward regulatory, tolerogenic, or inflammatory cells with unique characteristics to orchestrate immune responses. The development of monocytes into MODCs remains plastic until their terminal differentiation is used to acquire the mature phenotype, which is determined by a set of multiple extracellular signals like growth factors, cytokines, microbes, and metabolites, or the components of the diet. As you can see here, the vitamin A could be taken up by consuming foods containing its precursors like beta-carotene and in the form of retinol esters. Vitamin A, or retinol, is the immediate precursor to two critical active metabolites, retinol and retinoic acid, regulating the transcription of number of genes. Vitamin A does not occur in plants, but as we know, many plants contain carotenoids, such as beta-carotene, that can be converted to vitamin A within the intestine. So the retinol esters stored in kilomicrons and are transported into the liver, where the hepatocytes capture and store them as retinol. The molecule forms complexes with the retinol binding protein as you can see here, is the RBP and is carried through the circulation. The RBP retinol complexes after that are recognized by the retinoic acid 6 receptor, which initiates the absorption of extracellular vitamin A derivate into the cytosol, where the retinoic acid is generated. As you can see here, retinol oxidized into retinol or retinaldehyde by alcohol dehydrogenase, ADH, and after that, the retinol is oxidized by the enzyme retinal dehydrogenase, RALDH, producing retinoic acid, RA, which binds to nuclear receptors RAR and RXR, regulating the transcription of the target gene set by binding the retinoic acid responsive elements. These are the rares in the DNA. So the well-tuning regulation of DC differentiation genes is determinative in the appropriate modulation of lineage-determining master transcription factors, but which is more important in this project, additionally, ligand-dependent nuclear receptors have a major role in the regulation of myeloid cell differentiation as well, and could be classified into these visible four major groups of nucleic receptors. Inactive forms of nucleic receptors, as we know, are localized in the nucleus, repressed by coreceptor complexes or found in the cytoplasm, where heat shock proteins bind them. In the presence of their ligands, the nuclear receptors are released from the inhibition, and the binding to their specific response elements in the genome enables the increased transcription of target genes. So I think enough for this. Maybe it was too much detail, but sorry, I try to focus on. So more cell types has already identified like dendritic cells, macrophages, and stromal cells, which able to produce ATRA, modulating the differentiation process of sensitive cells like monocytes. Interestingly, in the gut lamina propria, the presence of DC subpopulations and MHCs could be detected, which cells could constantly express the enzymes for ATRA production, even in the absence of dietary vitamin A. Next, we analyzed whether MHCl cells, so our stem cell line, alter the immune regulatory potential of monocyte-derived cells via ATRA production. Thus, the expression of genes known to play the role in ATRA synthesis, namely RDH10 and RALDH1, 2, and 3, was examined in MHCl cells compared to the monocytes and differentiated MODCs. And as you can see here, we found that MHCl cells could produce ATRA based on our results. Next, the RAR nuclear receptors, as maybe we can accept this after this introduction, plays an important role in the regulation of specialized DC differentiation from the human blood monocytes. To examine the mechanisms guiding the modification in the phenotype of MODCs in the presence of MHCl conditioned media, we investigated the role of RAR using ATRA as an agonist and BMS as a selective antagonist of this nuclear receptor. For this purpose, as you can see here on this picture, in the corner, for this purpose, monocytes were incubated in the absence of RAR, in the presence, of course, of RAR agonist. After that, the media was refreshed, and the monocytes were differentiated in the presence of IL-4 and GM-CSF, and in the presence or absence of RAR agonist ATRA or MHCl-derived conditioned media. Based on our results, the ATRA and MHCl conditioned media similarly significantly upregulated the expression of CD86 and CTLA4. Additionally, the conditioned media increased the expression of HLA-DQ on the surface of MODCs, and as we can see here, BMS neutralized the effect of OTRA and the MSC-CM on the upregulated expression of CD86 and CTLA4. So the expression of CD86 and CTLA4 seems to be RAR ligand dependent. Thus, the possible presence of RAR ligands in the MSC conditioned media cause OTRA-like effects. That's why we wanted to examine the effect of OTRA on the phenotype of MODCs. So in the next series of our experiments, we inhibited the aldehyde dehydrogenase isoenzymes using highly selective inhibitor, which is the DEEB, in MHCl cells. In the following setup, the differentiation of monocytes was observed in the presence of OTRA deficient MHC cell-derived conditioned media, and of course IL-4 and GM-CSF. As you can see here, we found that the MHCl-derived OTRA has no effect on the expression of HLA-GQ molecule. However, the OTRA has an effect on the MHC-derived OTRA has an effect on the expression of CD86 and CTLA4 molecule on the MODCs. So these results suggest that MHCl cells bring about monocytes phenotypic changes, at least partially through OTRA production by MHCl cells. Finally, as the professional APCs, the primary function of dendritic cells to prime, activate, and polarize the T cells and T cell responses. To dissect the functional activity of MHCl conditioned media exposed monocyte-derived cells, ELISpot assays were used to examine the MODC-mediated allogenic T lymphocyte activation. We found that MHC-CM-treated MODCs can induce the polarization of the T cells secreting IL-17 and IL-10 at a significant level. To reveal the possible role of CTLA4 in MODC-mediated T cell polarization, the co-inhibitory molecule was blocked by specific neutralizing anti-CTLA4 molecule. And unexpectedly, the inhibition of CTLA4 signaling in MHC-CM-treated dendritic cells robustly diminished their ability to induce the development of IL-17 and IL-10 secreting T lymphocytes. Next, moreover, the flow cytometric analysis revealed that MHC-CM exposed monocyte-derived cells induced the polarization of IL-10 and IL-17 double positive CD4 positive T lymphocytes. Additionally, the generation of IL-10 and IL-17 double positive T cells was dependent on CTLA4 expression by MODCs. Because when the molecule was neutralized with anti-CTLA4, as was visible on the previous panel, the T-cell polarizing activity of MHCl-CM conditioned MODCs was significantly reduced. So our results demonstrate that MHCl cell released soluble mediators can regulate the T-cell polarizing capacity of monocyte-derived dendritic cells. Furthermore, the ability of MHC-CM exposed monocyte-derived dendritic cells to drive the polarization of these shown IL-17 and IL-10 positive helper T cell is dependent on the level of the CTLA4 molecules on their surfaces. So as a summary, in general, the MHCs, as we know, are awaking extraordinary interest among various stem cell types. They express a wide array of receptors involved in recognizing cytokines, chemokines, or invaders, endowing them to sense and react to different kinds of environmental changes. By the active communication, MHCs strongly interact with other cell types, including immune cells like dendritic cells or monocytes or macrophages, neutrophils, as I showed that in the introduction. The immunomodulatory capacity of the MHCs resulted in the inhibited proliferation of lymphocytes and suppressed inflammatory cells function after the activation. But in this project, we wanted to get to know the details in this regulation, or in their regulation, immunomodulatory activity. So furthermore, they can drive the differentiation of monocytes or the polarization of the T cell responses. Why we use the MHCL cells? Because one of the strategies to overcome the limitations of MHCs obtained from diverse source, culture conditions, phenotype, epigenetic background, proliferating capacity, secreted factors, is the use of MHC cell lines. Because these cells provide can give for us the possibility to generate MHCs with unlimited numbers of early passage MHCs with consistent quality and immunomodulatory properties. So in vitro generated MHC cells seems to be an appropriate model to examine the functional properties of the MHCs. In our project, we, as you know from now, we investigated the effect of MHCSS on the DC function, notably on the monocyte differentiation process. Based on our results, monocytes differentiated in the presence of MHCSS or MHC conditioned media into monocyte-derived dendritic cells characterized by a unique phenotype and functional properties characterized by or with upregulated expression of the CTLA4, as you can see that on the bottom of the figure. And this opening new perspectives in treating patients who have cancer, autoimmune diseases, or inflammatory diseases. The immune modulating MHCL cells indirectly promoted the differentiation of CTLA4 expressing dendritic cells by producing the RAR alpha ligand, for example, through, and as we shown, through all-trans retinoic acid, so ATRA. These monocyte-derived cells are potent activators of allogenic IL-10 and IL-17 producing helper T lymphocytes in a CTLA4-dependent manner. Our results suggest a new level of MHCs and consequently MODC control over T cell responses, and may have a remarkable implication for novel uses of the CTLA4 agonist and antagonist drugs. Mapping the molecular mechanisms of indirect regulation by MHCs on, for example, on the monocyte differentiation, we hope that could expand the clinical application of stromal cells in the cell-free therapies. I would like to thank your kind attention. Thank you very much.